This video is about pre-transcriptional regulation in bacteria or prokaryotes, and it is of course our old friend the lac Operon. If we leave bacteria in a sort of solution of glucose, you will get a graph that looks something like this, where you get bacterial growth sort of starting very suddenly, and it will trace along, and eventually it will level out as you use up all of your glucose. And if you've done populations and sustainability, you should be able to sort of talk about the different parts of that curve. That's not what we're talking about today. We're talking about what happens if you put it in a solution of a lactose. So if we move that bacteria from glucose to lactose, there's this really interesting lag phase where actually it's taking time to get going and entering that sort of logarithmic growth or exponential growth you'd see before. And this is basically people looking at why there is this lag phase and why it takes some time to switch on, to switch their metabolism to digesting lactose is the major. It won the people who did this discovery a Nobel Prize, and it's the lac Operon. So what they're doing in this time is they're actually switching genes on, so they're changing the genes they express in order to break down lactose. Making those genes usually would be a complete waste of energy and amino acids. We don't need them, so why waste energy and amino acids on them? But being able to make them on demand is very useful. And this was one of the first things of pre-transcriptional regulation, the regulation of what we are transcribing. So if we look at this is our bacteria with its lovely flagellum and its DNA, and it's got lactose on the outside of it, and it needs to somehow get some respiratory substrate. It needs to get some respiratory substrate to actually respire. So what sort of things are we going to need? We're going to need something to get it across the membrane, because this is polar, and then we're going to need something to break this glycosidic bond so that we actually get the glucose and the galactose out of it, because we can't just use lactose just on its own. So we're going to need to break that glycosidic bond, we need to get it in. So no point making those proteins now. We don't always need to break down lactose, but we do. And the two genes that we need to switch on are beta galactosidase. Oh, sorry. That is beta galactosidase. That is beta galactosidase, and that is lactose permease. So lactase permease is what allows lactose to go through the cell membrane, and beta galactosidase is what actually allows the breaking of that glycosidic bond to form our galactose and our glucose, which we can then use in respiration. So how do we do it? How do we go from not producing those two genes to producing those two genes, or producing those two proteins? And the answer is the lac Operon. And the lac Operon is actually this string of DNA. So if we look here, so I've zoomed in on this piece of DNA, and what we actually have is we've got lots of different regions to it. And basically you've got what's known as a promoter, an operator, and then gene. There's actually a third gene which we don't really know what it does, which we'll come to later, but these are the two genes for beta galactosidase and lactose permease, and we need to transcribe them. So this is actually our RNA polymerase, and it's not able to bind to its promoter, and it's therefore not able to transcribe those genes at rest. This is in a no lactose environment. The reason it can't is because further downstream on the DNA, just further away from the lac Operon, this gene is nowhere near the lac Operon, you have constant transcription of this repressor protein. So when there's no lactose around, this repressor protein will be made, and it will go along and it will bind to the operator region, and it will block the promoter so that no RNA polymerase can actually get on and transcribe those genes. This is how you stop those two genes being produced and wasting your energy. So how do you switch them on? And the answer is quite simple. Basically, when you actually get it, when you get lactose, and some lactose will be able to diffuse into the cell, okay, so some lactose will manage to get in at rest, but not a lot. Okay, what ends up happening is some will get into the cell, it will then bind to your repressor. So the repressor actually has a binding site, so the repressor protein actually has a binding site for lactose. it changes the shape of the repressor, so it can no longer bind to the operator region, which is this purple region here. That frees up the binding site on the promoter, which is where the RNA polymerase binds on the DNA, And then, basically, that RNA polymerase can now bind to our promoter and transcribe those two genes that we spoke about before. And we have now switched on. We have now gone from this state to this state. The third gene, just to put it into your sort of put it into context here in this sort of Wikipedia article, is the enzyme role in the classical E. coli Operon remains unclear. Okay, so that third gene you do not need to know. Even Wikipedia doesn't know.